So
hello everyone, thank you for joining our keynote talk. So we will have Christine Bacon here today to talk about her keynote talk will be parallel divergence across scales. And Christine is a senior lecturer in the Department of Biological and Environmental Science at the University of Gothenburg in Gothenburg, Sweden. And she has been working on palm diverse since her master's research on species delimitation in Mexican palms, and is now involved in developing a phylogeny of all species of Americas and examining ecological adaptation in Amazonian hyperdominant species such as Eutherprecatoria, Mauritia flexuosa, and Geonoma macrostachys. So Christine Bacon, thank you very much for accepting our invitation. And the floor is yours, or the screen is yours.
nice. Okay, can everybody hear me and see my slides? Can I get a thumbs up? Can you hear and see my slides?
Yes.
Yes.
Awesome. Okay, great. Well, thank you very much for this invitation today. It's a true honor to be able to speak to you, although I'm live from my bedroom. I'm Christine. I'm at the University of Gothenburg, and I wanted to, when I saw the program, take advantage of the nice basis that Hosani presented this morning and expand upon some other more population level work that we're also doing in Amazonia. So I thought I'd give a little bit of introduction, particularly for the students that are with us today, about some basic ideas related to divergence and speciation. And then I'm going to talk about Geonoma macrostachys, which is pictured here on the right, and talk about some evidence that we're slowly accumulating about genomic and niche divergence in this species. And then I'll move on to a quick section about morphological divergence and talk about next steps that we're doing related to this work. And so I've been working on tropical plant diversity now for over 20 years. So my first rainforests that I saw were actually in Brazil. And it's a shame that we couldn't be together in Rio today, but at least we can touch upon the exciting flora of that amazing country. And so my research focuses on tropical plant evolution. And one evolutionary process that's led to the high biodiversity that we see in these tropical forests is divergence, where populations of a species become isolated. They can become isolated ecologically, spatially, and then they can become eventually separate species. So it was Charles Darwin and his colleagues who first contributed some of these first works and ideas that were related to divergence in nature. And at that time, him and his colleagues proposed that natural selection was the main driver of diversification. Yet more recent works, mostly during the last century, and this idea of speciation and divergence was essentially viewed as the result of random processes, such as genetic drift, and that it was really these random processes that play an important role in the divergence of species. So following from the works of people like Ernst Mayr and Sewall Wright, where random genetic drift was contributing to the divergence of species. So this process is largely stochastic, where evolutionary change in the frequency of alleles or genes in a population are due to random sampling of organisms, where we can take, for example, as pictured here, different colors of marbles and make founder events in new jars. So this would be the process that divergence would go through if it was due to stochastic or random processes. And so in more recent years, really though, in the last 2 decades, the consensus view of this process of divergence and speciation has itself evolved dramatically. So now we find that the field itself is centered on identifying the mechanisms that are responsible for the evolution of reproductive isolation, especially when gene flow is still present during the process, when there's still crossing events between different individuals, populations, and species. So we can see this continuum of a focus on natural selection, a focus on random processes, and then more of a focus on particular factors related to ecology that's driving the speciation process. So this change in focus has resulted really in a renewed recognition of the role of natural selection, and in particular, ecologically driven selection as a powerful cause of speciation. Here I show two very important books that were published in the Knott years related to adaptive radiation and ecology, or ecological speciation. And so the potentially, though, more interesting side of speciation is actually just understanding the process of divergence itself, where a lineage can diverge with respect to genes or morphology or soil preference, for example, and that these different divergent factors can arise at different times along the continuum. So that's what I'm showing here in this excellent, one of my favorite papers by Kevin DeCuris in 2007, where he's showing that these different factors represented by these horizontal lines, it may arise at different time periods in the process of divergence. And what we've found in all of our research here in the POM community and biological diversity in general is that that because these divergent factors arise at different times along the continuum, they're not always congruent with one another. So we could have strong divergence, for example, in habitat preference, but little genetic divergence behind that. So when divergence is observed in nature, it's evidence for the role of natural selection. For example, when we have variation in beak size, it's hypothesized to be a response to diet, for example, in finches on Galapagos. So when divergence is observed, it's strong evidence for the role of natural selection occurring in different types of ecosystems. And when we see this type of divergence occurring in parallel, it's strong evidence for the role of natural selection and adaptive evolution. So first, I'm showing this process of divergence occurring in one cartoon where we have a common ancestor, this red circle, diverging into two. And when this happens repeatedly, we can see we have this other cartoon of this happening across, for example, this phylogeny. Or we can reflect on the nice work that's been done, for example, in stick insects, where in this phylogeny, we can see under similar conditions, the same morphotype evolves time and time again. So this process of divergence is occurring in parallel. So the repeated and independent evolution of these traits facilitates adaptation, and it provides one of the strongest forms of evidence for the role of natural selection and diversification. So there are different patterns that would suggest this parallel ecological adaptation, and it can result from different evolutionary mechanisms. But here we're going to focus on this third test case here, this parallel origin of ecotypes. And so there was this excellent paper produced out of Lauren Liesberg's lab that primarily works on sunflowers, that it reviewed the evidence for parallel divergence in plants. And what they suggested was that there are four particular criteria for parallel divergence, including independence across a phylogenetic tree, reproductive isolation between the most recent common ancestor and the descendants. Thirdly, we would have independently derived descended populations that may or may not have reproductive isolation. And four, that there's an adaptive mechanism or driver for the selection. So again, this is a table from that study that I just showed, reviewing the cases of parallel ecological speciation in plants, showing that in plants, little has actually been shown, but it's actually well known in animals. And we can see I've circled here one palm example that I'd like to talk about today, Geonoma macrostachys. And we can see that most of these citations in this reference list derive from the work of Julissa Roncal. And certainly, as Isaac Newton said back in the 1600s, if I've seen anything, right, it's by standing on the shoulder of giants. So there's been a large breadth of work by fabulous scientists in this system of Geonoma macrostachys. For example, Jette Knudsen has looked very carefully at divergence in pollinating insects. Robin Shasden, shown to the right of Jette, who is at the University of Connecticut, has looked a lot at light and adaptation to soils in Geonoma macrostachys. Julissa Roncal has dedicated a good part of her career to understanding these patterns. And now people like Maria Fernanda Torres are moving this work forward. What's interesting is that Julissa and I, as a funny anecdote, have been actually working on this system since 2003. I picture the two of us from our first fieldwork together in southern Peru in 2003. So if I have anything to say today about this interesting system, it's certainly because I stand on the shoulders of giants like these, and I thank them for all of their dedication. So the system I'll talk about today is Geonoma macrostachys. We know this to be a diploid organism. It has a large genome size of almost 2 gigabases. It's a small understory species that's widespread across the Western Amazon. Andrew Henderson, in his review of Geonoma, recognizes this as the most morphologically variable species in the genus, with up to 9 morphotypes that have been formally recognized, primarily based on leaf shape. On a field trip to Leticia, Colombia, I also have pictured here a photograph that I believe Rodrigo Bernal took on the different leaf shapes that we encountered in a single day of Geonoma macrostachys. So Julissa's previous work, I'm not sure how good this figure actually is showing, how good quality that image came out. But what Julissa showed in this 2006 paper is that in lowland forests in Peru, these 2 subspecies of Geonoma macrostachys were significantly different in their habitat preference, where one type was more abundant in the floodplain and one was more abundant in terra firm habitats. Listebarth from the Danish group also showed that 2 subspecies that differ in leaf shape, which is the focus of the work I'm talking about today, are reproductively isolated, and they are so based on phenology, flowering activity, and pollinator spectrum at a very local scale in this particular study. Julissa also included this work in her PhD dissertation, which showed ISSR, or some genetic marker variation that strongly partitioned sympatric populations where both these leaf types occurred together, that showed strong partition amongst these different subspecies. So that in 3 different forests that she was able to study, she consistently found this strong microhabitat difference that she showed in the previous paper. So this suggests strongly an independent origin of these different leaf types or subspecies in each environment. And furthermore, Finn showed in a special issue on palms that we had in 2016, strong differentiation between morphotypes at a particular site in Ecuador. So this segregation was supported not only by molecular markers, but also by leaf traits and data that we were able to get on floral scent chemistry. So this suggests, in this site at least, strong reproductive isolation at the local scale. So I've tried to give a brief introduction of this system. So to summarize, where 2 or more morphotypes grow together at the local site, they appear and behave as different species due to distinct morphology, distinct habitat preference, and potentially phenology and pollination. So these characteristics make Genoma macrosacches a really nice system to understand ecological divergence in Amazonia. So we began this study then with a first objective. We wanted to test these informal morphotypes that Andrew Henderson had recognized in his review in his monograph of Genoma using a morphological data set. So based on 29 morphological traits and a large amount of herbarium specimens, that I've shown mapped here on the left of the two different morphotypes, red and blue, meaning the two different morphotypes, we used some different analyses to statistically test the veracity of these different morphotypes. And what we were able to show is that our morphometric analysis yielded 4 clusters, which we can see here in the B panel. So as we expected, the 2 largest clusters were congruent with the most common and ecologically, or most common and easily recognizable varieties, I mean to say. So I show red here, we have this pinnate one, and these blue ones here is this bifid. So these 2 clusters then are referred to in this study, which we published recently, and that I'm talking about today, as different morphotypes. And these were used then, these clusters were used then to label the samples in a coalescent phylogeny, in analyses related to habitat differentiation, and on reproductive biology. So we also hypothesized that representatives of these different morphotypes would group geographically, but not necessarily by morphology. So our goal here was to look at the phylogenomic relationships and genetic structure of this species sampled throughout its distribution. So we sampled individuals across its distribution, 43 of them, using target sequence capture and various different analytical approaches. So although this coalescent species tree should be interpreted with caution because of the large parameter space associated to it, so this is a very large data set, not necessarily regarding the number of individuals in the phylogeny, but the number of polymorphic sites analyzed from this target sequence capture experiment. But what we are able to see in the upper left-hand corner is the posterior distribution of the trees is suggesting high gene flow. We can see that in the upper left-hand corner that looks like a cloud of branches. So this is suggesting either high gene flow and or incomplete lineage sorting. When we reflect on the species tree that we reconstructed, we see that these morpho groups, or these leaf types, again, differentiated by the red or the blue, are not monophyletic, and they don't match these clusters that we found based on the morphology. And so as then we had, as we found here, the morphologically similar individuals did not form any groups. So this decoupling between morphology and genetic structure maybe could be the result then of some sort of ongoing divergence or speciation with gene flow, or it could be hybridization after secondary contact of these divergent morphological types. So we also wanted to not only look at the phylogenomic relationships, but also the genetic structure of the samples that we had from across the distribution. So we again hypothesized that the representatives that we sampled would group geographically and not morphologically based on the phylogeny that I just presented. So here, this is just a population structure analysis where we're trying to identify the different genetic clusters present in our sample. So here, the most probable number of genetic clusters that we found was 4, which support, and these clusters supported 2 main groups. So we found this widely distributed genetic cluster that's occurring in the north and south of the study area, this brown one. And we found a group in the southwestern Amazon, which we can see here in the green, this dark green. These 4 genetic clusters do not correspond to the 4 morphological ones that we found that I showed previously that were observed by their geographic locations. So here then we're showing that it's geography rather than morphology yet again that is forming the clusters here. So based on morphology and on genetics, we're seeing that it's geography rather than morphology that is uniting these different samples. So our third objective was to look at habitat specialization or differentiation in this group. So to test for habitat divergence, we merged both climate and soil data. So we had information about soil chemistry based on Henrik Balslew's transect data set. And then we also used information on climate from BioClim. So we had a large amount of transect data and we had lots of sampling points. And we decided to take this at 2 different scales. We were interested in divergence at the continental scale, so across all the samples in the Western Amazon. And then we wanted to look more regionally. So we took our best sample population in the Napo region of Peru to look at a more regional scale. So based on the suggestion then that morphotypic habitat preference it may or may not be consistent through its distribution, we didn't expect to find complete habitat segregation at the continental scale, potentially at the regional scale, but not at the continental scale. So here, when we look at our results of niche divergence, or the climate plus the soil, we find very interesting results with respect to local adaptation. So here I'm plotting habitat variation as a proxy for niche-based differentiation on these 2 different sampling scales. You can see here in panel A is the continental scale, and in panel B, and what you can see the little map, is the more local scale. So in red are transects where only one of the morphotype occurs, so this echoless morphotype. And in the blue, again, is the other morphotype, the bifid one, the macrostachys one. Those are in what you could call allopatric populations. I'm also plotting information from transects where both of the morphotypes are occurring together. That's what we see in this green cluster. So it's where the 2 morphotypes co-occur. So at broad scales here in panel A, what we found was that niche differentiation is certainly incomplete at the continental scale. Yeah, we can see that that niche divergence or niche preference is largely overlapping. But when we reflect then on panel B, we can see that there's strong niche differentiation at this local scale. So we can see them highly segregated. So when we take this together, we interpret this as strong evidence for ecology as being a fundamental driver in this system of divergence, at least at local scales. And we also had the objective to look for reproductive barriers between these different morphotypes. So we wanted to look at pollinator guilds and phenology between these 2 different forms at our site in Leticia, Colombia. And so under this hypothesis of ecological divergence, we expected to find reproductive barriers throughout the distribution of Genoma macrostachys. It had been suggested, or it had been quite clearly shown in Yasuní in Ecuador. And so we had expected to see similar patterns here. So when we had our 3 field campaigns and we measured nearly 100 individuals for these different factors like flowering synchrony, antithesis time, flowering rhythm, stigmatic receptivity, and I think it was pollen and pollen viability. And so what we found, though, was a time lag between these morpho groups regarding the time of daily anthesis. So male flowers of the macrostachys form was reaching anthesis earlier than those of echolus. There was an overlap period, but reproductive efficiency was significantly lower in one than the other as well. And But we also found that the diversity of insects visiting the inflorescences of these two different morpho groups or types was essentially the same. So most of the visitors were using the flowers for food, mating, and oviposition, but the diversity was largely similar. So we found not very strong reproductive isolation between these two groups, at least at a local scale. So when we take this together, all of this evidence that I'm showing to you now about genomic and niche divergence in genoma macrostachyes, we find that these morpho groups are not monophyletic. They don't match the genetic clusters, and the spatial scale of habitat differentiation was more evident at the more local or regional scale than at the continental scale. We also found no evidence of prezygotic barriers at this very local site in Colombia. So here we want to then entertain different hypotheses of what would drive this leaf variation in genoma macrostachyes. We know that phenotypic plasticity is very common in plants. So, and plasticity can occur at different levels. And for example, I'm showing here, we've got the same species growing under different light conditions, and you can see drastically different leaf and individual variation here in this polygonum species. And it's very common in plants, phenotypic plasticity. It's difficult to distinguish phenotypic plasticity from adaptive responses. So, Mia, no. So, excuse me. So, another alternative to explain the variation in leaf shape is phenotypic plasticity, which has never been tested in genomic macrostachyes or even in palms because it's very difficult to do common garden experiments. So we had tried to do this in genoma after our field work in Leticia. You can see here our colleague at the University of La CES in Medellin planting out the seeds that we collected from Leticia. Unfortunately, we had very low germination, and then Corona happened. So the university was closed, the greenhouse was closed. So this experiment has now been abandoned, unfortunately, but we do hope to go back to it. But we do suggest that phenotypic plasticity is not likely, right? So different genotypes underlie two local morphotypes that are specialized to these different habitats. For example, in the Yasuni experiments and in Loretta, Peru, from Julisa's work. So while we can't reject the hypothesis about phenotypic plasticity, we do believe that it's unlikely. So now I'd like to move on to another part of this genoma macrostachyes work that's related to morphological divergence. So while our recent publication is suggestive of ecological divergence of these different morphotypes, there's a lot more work to be done, as always, right? So with funds from the Swedish Research Council, Maria Fernanda Torres and I sampled densely within a single site in the Colombian Amazon. Here's a picture from our trip in 2019. So in the field, we collected from different populations of genoma macrostachyes, including those, again, where one single morphotype is found, and populations where those morphotypes co-occur. So that's shown there in the middle. So these collections allow us to perform what is called whole genome sequencing, or low coverage whole genome sequencing. What we do is we have a pooled approach where this is a population genomic approach that allows us to test for local adaptation of these different leaf types, where the 4 different pools that I'm showing you here allow us to look at the morphological divergence of these different leaf types. So here we have sampling from populations, again, where the morphotypes are found uniquely and where the morphotypes co-occur. So this approach is really letting us get very deep DNA sequencing from full genomes and maximizing the sampling of rare and potentially adaptive alleles, which we were unable to show in the previous study. And so this is a figure that Mafe prepared where you can see the different actual spatial, don't know if you can see my cursor, but this is the actual spatial distribution of these different pools that we did. And the kind of hypotheses that we can test from that. So our goal then to test for local adaptation using these genomic data is to look at genes that are linked or directly under natural selection. And we do this by generating large amounts of SNP data and positions of variation across the genome, and looking for those loci that have genetic diversity values, or FST, as I'm showing here in this cartoon, that are outside the expectations from Hardy-Weinberg equilibrium, which is the expectation of neutral evolution. So random mating, no migration, no gene flow, etc. So these loci, these outlier loci, they're referred to, are tightly linked to or directly under natural selection. And although they're rare in the genome, which we show by this distribution of frequency, they can reveal important functional genes that may or may not be, right, responsible to adaptation in these contrasting environments. And so this is just some very preliminary data that we have based on this pool-seq experiment that Maria Fernanda has been working on. So here we've cleaned and filtered the data on different types of values, and we can see that my kid's totally freaking out in the background, sorry. It's a tricky time of day. And so we are able to find a small suite of of these outlier loci. And what's interesting is we can then map them, or we can, you could say, like align them against, or assemble them, map them is what it's called, against a gene ontology database. So genes that we know based on Arabidopsis, et cetera, other important plant model systems that are important for leaf and flower genes, like Knox, like these homeobox genes, to look for interesting SNPs in Genoma macrostachys. And while this is very preliminary, what we were able to find initially were a handful of SNPs that may tell us why these morphotypes are genetically different beyond some value of background variation. And that these differences may be sitting somewhere important on the genome. So we can then annotate based on our reference genome. It's important to note that these methods, and in general, any type of genome-wide association study such as this, may identify strongly differentiated loci. They're expected from these monogenetic traits. But we definitely are going to lose power when we're looking for polygenic traits, or morphologies that exist because of multiple genes interacting. So the more genes that are involved in diverging these leaf forms, the less power we may have using this type of approach. Moving on from what I was able to show from the SNPs and the outlier loci, we have received long read sequencing, and then we complement that with Illumina short read data to create a new and improved reference genome based on this nanopore data. So this is going to improve the power that we have to identify the genes responsible for these different leaf types. So interestingly as well, that I did want to mention in case other people are interested in long read data, is that we got fairly low throughput in our experiment due to blocked pores. And although we were able to wash various types of nuclease and reuse the flow cell, we definitely had issues. So I'm interested in any of you have had similar experiences or have any suggestions on how to improve future experiments. So with this, we will identify patterns of genetic divergence and morphology across the whole genome once this reference, this new Genoma macrostachy specific reference is ready. So maybe there are strong differences, but somewhere else in the genome that we haven't analyzed yet, that now we will be able to. We also want to expand to get more at this kind of parallel nature of the system that I began this talk about. And to do so, we're interested in sampling multiple individuals from more populations across the range. So here's the map from our paper from this year, and you can see that there's a lot of populations that we can sample. And it would be interesting to do this low coverage genome approach to actually explicitly get more data that would allow us to get at the gene flow hybridization aspects of this. So I may have gone from the very broad to the very specific, but I think that understanding how and why variation arises in nature is really important. Advances in genomics and biodiversity science are especially important in forests such as these in Amazonia, yeah? So recent media coverage of the fires, particularly in Brazil, has increased public awareness of these strong land use changes throughout the continent. And we know that even in 2019 alone, that more than 1 million hectares were lost to fires, and most of this was occurring in Brazil. And this is important not only for biodiversity per se, but it's coupled with the fact that vegetation isn't just a silent actor in climate change effects. We know, it's actually unequivocally known, that forests have a strong feedback with climate and can change it independently through the hydrological cycle. And this has even prompted suggestions of this like approaching tipping point, yeah? And in Amazonia and the Arctic, there are other places where these tipping points exist, but the loss of the Amazon rainforest is certainly one of them. So this raises the question then of how much deforestation would be required to cause this cycle to actually degrade to the point of being unable to support rainforests. So we know that rainforests are fundamentally important, yeah? So they hold a disproportionate amount of biodiversity per square meter on the Earth's surface. We can see here in this map from Kreft and Jets, the lighter colors are the higher amount of biodiversity. We also know that they have the highest values of global natural capital. So if we actually put like a dollar sign or a dollar value to each hectare of land, the highest values seen here in the more red colors, warmer colors, are found in tropical areas. But the issue is that it's these exact regions that have the highest forest cover, which are shown here in green, are also the ones that are experiencing the highest level of forest loss. And this idea of habitat and species loss is important for society and for us here, because not only do we maybe have a moral responsibility to maintain this diversity, but humans also depend on it. And I'd like to suggest that this is exactly why what we're doing, observing, mapping, measuring, sequencing, modeling, etc., is so very important. And so I just wanted to say to keep it up, and Viva Palms and Viva the Palm community. I took a couple pictures that I had on my phone from past conferences and fieldwork and such. And I just want to congratulate you all, because I think that we are doing our part, but we do need to do a lot more. And I think that the efforts that we're making through the IUCN committee is very important. And so, yeah, I just wanted to look forward to PALMS 2025 and UNOPS 2022. And I also wanted to thank all of you in the community, and one member in particular that I wanted to congratulate. My friend and collaborator, Andrew Henderson, is retiring next month from the New York Botanical Garden. I have a picture of him below looking in a microscope at Linonia specimens when we were in the process of recognizing a new genus of palms from Vietnam. And in the picture above, we have him reviewing his notes on, I think it was Cocothrynex. So we've had so many wonderful conversations, and we continue to work together now on a review of hybridization in palms. And I just wanted to remind you all and congratulate him on his long and very palmy career. And so I'd like to thank you all for your attention and the great honor of presenting our work here today. So thank you very much.
Thank you, Christine. Thank you very much for your very inspiring talk. And so we have a couple of questions here. And I also would like to say thank you for giving this talk in not a very convenient time for you. I know that it's sometimes hard to conciliate everything that is going on in our life with opportunities and things we want to do. And so I would like to extend my special thanks for this too. So I will start with we have some questions already, and feel free to post some more. So the first one is from Cynthia Freitas, and she said, great overview of the results, Christine. I wonder what makes genoma so morphological variable, comparing, for instance, with Uenocarpus, Socratia, Euterpe, and other Amazon genera. Do you think might be related to the fact it's understory species, small size, or maybe related to generation time?
Yeah, I mean, I would need to reflect on generation time, but I think you've touched on the important aspects of it being in understory palm and the distance that pollinators and seed dispersers are functioning under. So I think you get a lot more specialization in understory palms than, for example, Enocarpus or Euterpe. There's a lot of species complexes such as this, other ones in genoma and other genera in the understory. So I think this is related to pollination and seed dispersal.
So there is one question from Rosani Colevati. Christine, congrats. This is a great and beautiful work. Thank you for sharing your results. And now she has a question. Have you performed transplant experiments in the field to verify whether morphological differences are due to phenotypic plasticity?
Yeah, so I did touch on that. Thank you for that question. So we did not try reciprocal transplants in the field. What we tried was a common garden experiment. But unfortunately, due to multiple reasons, so that the only one of the morphotypes had mature seeds or fruits at the time, and then we had low germination rates, and then we had greenhouse closure. So it was a failure. But this is something we want to do, when we can get back to the field. Yeah.
And question from Rita Portela.
Yeah, you want to come? Yep.
Is she coming?
Yeah, she's coming. You want to say hi? Oh,
yeah. Better now, huh?
Yeah.
You want me to read? Yeah.
Yeah, oh. Yeah, go ahead. Yeah.
Christine, you should be very proud of your mother, Mia. Christine, thank you for the wonderful talk. Just a comment and an invitation. You should come to Atlantic Forest and study the Genoma Polyana subspecies. There are 5 subspecies occurring together, and in some plots, size 30 to 10 meters, we can find 4 subspecies together, but all of them in very low abundance, so with very low fruit production.
Yeah, low abundance is very strange. There's lots of systems like this. So Maria Jose Sanin has also done a lot of beautiful work on the, I can't pronounce it very well, Orbigniana, the Genoma Orbigniana group in the Andes. So there are many groups like this, particularly in genoma. So it's, I agree, Heath, that would be very nice to do. But again, like Cynthia was saying earlier, it's very expensive, and all of the work and effort that it takes, and the samples that it takes, and the investment. So I think there's many interesting systems, but I think a good approach is also just to try to focus on one system. So I don't have a lot of students, for example, in Sweden. So I'm trying to focus on that one system so we can really make an advance. But I agree, the Atlantic forest has some interesting complexes as well.
So I think I have a question myself. So you look at this large scale regional like distribution and the small scale but can you do you have a guess about what are the environmental conditions so that could be underlying the morphological variation or things like that? Because when I look at when we think on geography could be two things at least. So it could be difference in space, but could also be differences in the environment that are just happen to be represented in space also. So do you have a guess on which environmental conditions?
Yeah, I think it's interesting. I don't think about strata space, no, but I think well, Julissa has shown it extremely clear that habitat preferences it's completely different, right? So one is on the Varsia, one is on the terra firme. So there are places where they co-occur, but there is strong habitat preference differences. But I can't say about, whether one might be a bit taller or whatever. I don't think so there. But I mean, there are strong habitat preferences. Yeah, and that's what we show at not at the continental scale, but at the local scale.
Yeah, this is very interesting because I have been sampling like macrostacks for a while, and I could not, like, from my field experience, tell what could be like where to find one or another. This is very interesting. So we have a lot of questions, and so one from Tomakuver. Great talk, Christine, and a nice overview. I shall be looking at, sorry, we start again. I shall be looking at macrostacks with different eyes now that when I'm in the field. Do you think structural variation at the genome level could also be an important factor linking to the incredible variation in other genomes too, for example,
structural variation would be very interesting to look at, but again, we need more genomic information. So even with long read sequencing, well, I mean, now we have a reference genome, but for structural variation, you'd almost need chromosomal level information. So we're not there yet in genoma, but we would like to. And I think that's an excellent point, Tomas, that structural variation is increasingly being shown to be important in divergence.
Yeah, and he suggests this would be nice to look at nanopore data too.
Yeah, we use nanopore for the new reference for genoma macrostackies, but we don't have nanopore data at the population level for population genomics, no.
There is also another suggestion from Rosane Colevat on techniques. So Christine, just a suggestion, I use Sweep Finder to seek for selective sweeps in high hitchhiking in genes leading to divergence between morphotypes.
Yeah, it could be interesting. But again, our better data now is it's pooled data between different pools of populations. So we have allopatric macrostackies, allopatric and acaulis, and then we have the individuals from sympatric populations. So we lose the individual level in that pooled experiment. But yeah, we need to do more sequencing. Yes.
So I would, since we have a lot of questions still, I will try to address first the ones that haven't asked it before. So sorry if you have posted multiple questions. I will ignore you for a while. Great talk. A question from Bill Baker. Great talk and masterful navigation of the parental minefield. Very good. Wonderful to celebrate Andrew Henderson's amazing career. I will talk a little about his stellar achievements on my talk on Friday. looking forward. What do you and people doing your kind of work need to future POM taxonomists to support your work? So how do you see our work feeding back on taxonomy, your work feeding back on taxonomy? Sorry.
I mean, I think that we can't protect what we don't know, and a law or an entity cannot function unless there's a name, right? And naming is taxonomy. So then what we are doing is leading to names, which is leading to taxonomy. So the IUCN can't do anything about anything if it doesn't have a name, you know, and they need to have right names. So I think that's how what we're doing feeds into taxonomy. So this is a small window of the work that we do together with you, Taizi. We're also recognizing new species from Amazonia. So this is a small portion of the research that we're all doing together as a community. So taxonomy is the basis. And I think that, yeah, that we will not be able to overcome this biodiversity crisis if we can't appropriately name things, map them, and categorize them. They need us. Society needs us. And so we need to keep doing our work.
A question from Maria Cristina Penuela. We are at the Young University Regional Amazônica ICANN. I don't know if I say it right, in Ecuador. And have the Colonso Chulupas Reserve as our laboratory, 3 kilometers from our campus, so so we can perfectly work with students here. You are very welcome. I think it's a comment and an invitation. Question from André Silva Pinheiro. Congratulations for your talk, Christine. According to what you said, genome is a good model to understand the genetic and morphological patterns that undergo during leaf development in palms. I guess there are no works that analyze these issues in genome. This is surely an interesting research. And I don't know if you want to comment on that.
Yeah, I think looking at the leaf development in genome is extremely interesting. It is not per se within my expertise. But I think that there's a lot of capable people that could be dissecting lots of apical meristems and all sorts of meristematic tissue from leaves. I think that there's a lot to be learned. There was a talk earlier today by Maria Fernanda Torres looking at leaf shape and how leaf shape, what drives variation in leaf shape. So I think there's a lot to be done. I don't think macroecology is necessarily the approach to do so, but it is an interesting one. And I think it is a lot of anatomy could also be interesting.
A question from Bruno Medeiros. Great talk, Christine. Genome, a lot of congratulations. That was a great talk. Is genome macrostachys used by human populations in any way? I'm wondering if the phenotypic traits that vary between morphs could have any impact or bias on biased dispersion or extraction mediated by humans.
No, I don't think genoma's macrostachys is interesting to. You mean use by humans? I don't think so. No, I think that. They are so few, they're not very abundant or common, and their seeds are not, they don't ripen all at once, they're fruits. So maybe they would, I mean, other people would know much better about the ethnobotanical significance of Genoma macrostachys. Maybe Rodrigo Bernal has something to say about the uses, maybe in some sort of handicrafts, but I don't think there are very many uses for it. So I don't think that it's a human impact issue there.
So we have 2 more questions, and I think then we can close. So another question from Cynthia Freitas. Christine, this is so cool. This is a so cool setting regarding genome individuals with different morphologies. Do you think it's going to be a good example to test ecological character displacement?
So character displacement. I mean, potentially, but we would need, much more sampling. I think there's a lot of place for something like geometric morphometrics, where you're using landmarks and these types of things to actually explicitly test that type of thing, which we are not doing, but that is a totally open field. We're doing that with a CAPES funded project in bromeliads, for example, leaf shape in bromeliads. And I think that there's some interesting things related to character displacement, but you would need the right data for that. And I think that something like, 3D morphometrics could be quite interesting. But I think, Cynthia, that's an interesting suggestion.
A question from Li Xiu Gu. Sorry if I didn't say it right. I think it's more a comment. Great talk. Hope there will be a chance to work on Chinese species. I think that everyone wants you everywhere, Christine.
Yeah, there's a lot of interesting Linonians and koalas in China. Yeah, that would be good.
Yeah, so speaking more on this thing on so collections are needed and people are needed and more taxonomy are needed and everything is needed, and Cynthia make a question here more into diving into politics and how we can press governments such the one we have in Brazil right now on the economic importance of a biodiversity. So I feel that we have these two, like a force. We need to collect more and do more at the same time. Things are getting more challenging in terms of resources and the speed we need to sort those all this. So do you want to say some final words on how do you see we can do more with less and fast as a big example of someone that is.
I mean, I think as women we're already doing that. You know what I mean? You said to do more with less, right? It's like, but I mean, I don't want to be political at all. Maybe I've been in Sweden too long to want to say anything overly political. But I think that very soon we will reach a point where it's no longer a choice, and it may take some countries longer than others to realize that. But I think that the economic recovery after Corona will largely square on climate change. And that is, we're going to see that in the European Research Council calls. This is the only way forward. And if there are some countries that are lagging behind, then we all need to help them. It's like sharing vaccines. We need to share resources to get us out of this terrible mess we're in. Yeah.
So
I don't know, but that's not a good way to end. That's not a good way to. But I mean, yeah, it's as I showed in my picture, I think there is 70 people or so that still want to listen to this conference today, amongst all of the work and social and family that we are committed, and we are only a small drop in the bucket of people that are doing what we're doing. And so it's important that we keep doing what we're doing and keep training new students. And I'm continually so impressed with the students that are arriving to work with us and study with us. The quality is ever increasing. Our knowledge is growing. It's amazing. So I think that there's a lot of reason to be motivated and reason to be inspired, because like I said, we're standing on the shoulder of giants. We're building and we're going somewhere. So I think that that's a good way to end on a positive note. Yeah.
Good. Better. So I would like to thank you, Christine, again for being here with us. And so this is the last talk from our live program today. So we are very much looking forward to see you all again tomorrow, and will be 9:40 Brazilian time with the question and answer for ecology section. So as you You may know by this time all the talks are pre-recorded, and the posters are there also. So have fun and see you tomorrow again. And so bye bye. Thank you, Christine, and thank you everyone for being here.